Hello everybody, and today I'm going to show you how to use a hemocytometer for single spore isolations and serial dilutions. Hemocytometer is used for blood cell counting. Hema or hemo meaning blood, cyto meaning cells, and meter meaning measuring. So it has two little sections on it. So you can use it twice once it's clean. And it has two, on each one it has a tic-tac-toe pattern about. You can kind of see it. We'll jump over on the microscope here pretty quick and get a better look. So regularly, if you're using this, everything would be sterile. The flow hoods would be on. We'd be working with sterilized water. I'd be all cleaned up. I'd have a mask and a hat on and everything ISO'd down. But my flow hoods are too loud for the video, and I'm not going to really use these spores in projects. So really just going to do this for the sake of the video. Yeah, we'll just go ahead and get started. I'm just going to use some regular old water from the sink. Again, everything's just done. I'm not going to use these dilutions. Regularly, I would have sterilized centrifuge tubes, sterilized water, a sterilized solution of tween 20, which is this stuff right here, polysorbate 20, and it's pretty much just a detergent, so it keeps your spores from sticking together. I find that with fresh samples, fresh solutions, that it's not really a problem, but if you let them sit for a while, then it definitely is. So that can be really helpful and absolutely necessary for single spore isolations. Sterilized syringes. I like to use syringes. I find that they have the most accurate drop size compared to other droppers. These guys are nice because they're completely autoclavable. So are the syringes though. But these guys aren't usually. These will both work also. I still like the syringes. I'm going to get into these a little bit more in a minute, but micropipettes, these are crucial, especially this guy right here. The smallest micropipette you can get is a 1/10 of a microliter to 2.5 microliters. So, and we'll get into that in a second and see why that's really helpful for when you're using these hemocytometers. They have sterilized pipettes, so that kind of just makes working really easily. Centrifuge tube holder. Microscope slides. I usually have sterile ones that I use in sterile conditions. These ones are not. And yeah, we'll just go ahead and we're going to load the hemocytometer up. We'll look at it under the microscope here pretty quick. Yeah, let's just take a quick look at it actually. So here we're all centered out on one of the sections. Kind of close up. We'll go back real quick. Oh, no, that is all the way back. Okay. Whoopsie. Alright, so this is the center section on one of the... I'm going to post links for better videos on how to use a hemocytometer. I just think that it'd be helpful to kind of have a spore-related one. So this is going to be your first 16 square section, one of the 4 sections that you'll be counting. You only use the 4 corner sections that contain 16 squares. So that's one of them, 2 of them, our third, and our fourth. So pretty much how we're going to fill this up is we're just going to put a cover glass slide on it, and then pipette a small amount of liquid into it. So how you fill one of these things is you're going to take your hema and just put it on your microscope. Get a cover slide. Go ahead and place it right centered in the middle. And what it has is kind of like a flat surface with edges on each side and the cover glass goes over it. So there's a little section in between. And the space of that section is 1/10 of a microliter. So what we're measuring is actually the same size as the smallest setting on the smallest micropipette. So, and what we're doing is we're counting each section that we're counting, each of those 4 sections of 16 squares is 1/10 of a microliter. So we're going to get an average for those, and then times that by 10,000, or 10 to the power of 4. And then that's going to give us our average spore count per milliliter, because a microliter, or 1/10 of a microliter, the space that we're measuring on average, is 1/10,000th of a microliter. So let's get a little solution real quick. Since everything's not sterile, what I'm doing is just taking this jar of water, and I'm going to take the syringe, take like 5 cc's, and put it in the jar lid. And take our spore sample, spore print. This is a P.O.P.N.Y. spore print. We're just going to get a scalpel, scrape a little bit onto the tip. You won't be able to see it in the video, but it almost looks like I just scraped up a little bit of dust or something on there. We're going to mix that right into the water. You may want one of these things. It's just like a mixer pretty much. So you push down in the center and it just vibrates and spins whatever you put on it, whether it's a spore syringe or a centrifuge tube or really just about anything. We aren't really going to need it today, but I'll probably show you that in another video. So we have our spore solution all mixed up. What we're going to do is take it back up into the syringe. Pick it all back up in there. Right. So you have to do these really carefully. You pretty much just take a drop on the tip of your pipette and put it right up next to the edge of where the hemocytometer meets the glass slide, and it kind of just feeds in there because there's space for it to go into. You want to make sure it's nice and full. A drop is about 50 microliters. The slide holds about 20 microliters, so about half a drop. Drops will vary. So let's try to get back up there and see what this is looking like. Oh yeah, look at all those. A lot of motion. A lot of motion. Start to see the plane they're on. They're moving a bit slower. Making sure there's no water on top of the glass. A lot of movement there, but I think it's slowing down. Okay, that's looking pretty good. So let's center it back out real quick. Seeing all these spores. Take a little closer look at these guys before we start counting. There they are. Happy little babies. Alright, let's go back. So what we're going to do is count the total spores in these 16 squares as one section. These 16 squares is another. These is the third and the fourth. And each of those 4 sections is measuring 1/10 of a microliter, which is 1/10,000th of a milliliter. We're going to take our average from those, and it's going to help us do our math. So we'll go through and look right here. 1, 2 in the first cell. Let's get a little bit closer. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33. We'll just say that's a rough 33. One, 2, 3, 4, 5, 6, 7, 8, 9. Call it 10. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. 26, 27, 28, 29, 30, 31, 32, 33, 34. Yep, overcounted. It's 31. 32, 33, 34, 35, 36, 37, 38, 39. Call that 39. Okay. Now I'm not going to count all these because it's really not important. What's important is that you get the idea of how to use this thing and understand how much easier it could make your life. So let's say we had 39 and 33, which is 72. The average of 72 would be 36. So let's just say we counted all 4 of those squares and we got 36. So we would take 36, which is our average. So, I mean, you would times that by 10 to the power of 4, which is 10,000, just for the sake of simplicity. Some people think powers are more simpler. I think it's kind of confusing. So, with that said, it would give us 36,000. Yes, 36,000 spores per milliliter. So in just 1 milliliter or 1 cc is 36,000 spores. So if we wanted to dilute that in half, A really simple way to think about that is just a 1-to-1 ratio with this to plain water. That would take it down to, what's half of 36? 18 spores per 18,000 spores per milliliter. And if you look at it like that, you could divide it by 10,000 to find out how many spores per tenth of microliter. So say you watered it down to half and now you have 18,000 spores per milliliter. That means that you have 18 spores per tenth of microliter. So you're getting pretty close to the point where every drop from this guy would have 18 spores. I mean, you don't have to do much math before you're at the point to where it's just one drop of spore. One drop, one spore. And that's kind of what I'm getting at with this video, is how you can really utilize a hemocytometer and this specific micropipette to really easily do single spore isolations. I'm going to link the videos to how to more appropriately understand using the hemocytometer, but also an article that I found that kind of put me on to the hemo in conjunction with this specific pipette. It pretty much turns into a probability, because obviously we're doing our math on averages. So you're going to, if you really dial it down to where there was on average one drop per 1/10 of a microliter, then you're going to have some drops that have 2 or even 3, and then some that have none. But on average, they should have about 1. So you're going to have a lot of potential for monokaryotic growth with that. So I'm going to kind of pull up this whiteboard for a second and do a little bit of math with you guys. Hopefully it'll help everybody visualize. Well, let's say we had on average 5 spores. Oh yeah, there's no way it's going to be able to be seen. And some people count the center square. It really doesn't matter. I mean, you can. It might make your average a little more justified, but you can do 4. No matter what you do, if you're counting 4 or 5, make sure you just do your average. You divide your total by 4 or 5. You'll see that in the other video I linked. I'm just really trying to simplify the process. It's just, you could do a lot of math, or you could just make it a little bit easier. So let's say we have an average of 5 per cell. So we're going to take 5 times 10 to the power of 4, which is 10,000. And that's going to give us our total per milliliter. So that's 50,000. Right? So we kind of went over that already. But what if you wanted to dilute it down to, say, an average of 1 per square? So how you could do that essentially is a 1-to-4 dilution for this specific example. So say you had one drop that had 5 spores in it. If you diluted that down with 4 other drops, then you would have 5 drops with 5 spores in it. So you can kind of look at in different ratios to get you closer to that 1 that you need. So if you did 1-to-4 dilution of the above solution, that would give you a average of 1 per cell, which would look like that. So if you took your average of 1 per cell, 1 times 10,000, because remember, each one of these squares is measuring 0.1 or 1/10 of a microliter, which is 1/10,000th of a milliliter. So, starting to make some sense to somebody, hopefully. Okay, so say you don't have a micropipette, okay? And you really want to do spore dilutions, and all you have is just like a standard dropper or something like that. Well, we talked about how a standard drop is 50 UL. And we made our ratio over here for 1/10 of a UL. For 1/10 of a UL. Microliter. Sorry. Now if we wanted to get it diluted even further to the point where there was only one spore in a standard drop of water, which is, 1.1 microliter is 1/500th of 50 UL. So what we would need to do is take a single drop of this solution. So if you can use a hemocytometer and dilute it down to 1 spore on average per each one of these squares, that'll give you the solution of 1 spore, or I'm sorry, 10,000 spores per milliliter. That's this solution right here. So if you wanted to take that and dilute it even further for a single drop, you would basically dilute it to a 1 to 499 solution. So it will give you 1 drop to 499 parts water, which pretty much is, when you dilute it similarly to this. So if we did 1 to 499 of this is water, and this is the 10,000 per milliliter solution. What that would give us is 1 drop to every 50, or 1 spore to every 50 microliters. And there you could just use a standard pipette or dropper. So hopefully that makes some sense to some people. Real quick, I was thinking how you would do the math for that above solution. So what you would have is, bring this back real quick. If you did say dilute your solution to that far of a rate, to where there was 1 spore to a standard 50 microliter drop of water, I guess what that would look like is, because right here we have 1 to the power of 10, or 5 times 10 to the power of 4, and then it goes down to 1 times 10 to the power of 4, 10,000. So what we would have is 1/500th times 10 to the power of 4, and God, it's just slipping me right now. What is that number? Let's see if I can do that math real quick. Yeah, that's right. I had that figured out before. Okay, so if you took your 1/500th, which is the equivalent of 20% of 1. I'm sorry. It is 2,000th. It is 2,000ths of 1. Okay. So that would actually give you 20 spores per milliliter, which makes absolute sense because there's 1,000 UL per milliliter, and if we divided 1,000 milliliters by 20 spores, it would give us 50 microliters, which is a drop. So you would have 20 drops, 20 spores, so hopefully that makes some sense. I know that this whole mushroom thing is moving on very quickly, and you know, I guess for the sake of the video, you know, if you were gonna do if you're gonna take this, what did we say? We had about 36,000 per milliliter. So we have 5 milliliters in here. 36,000 times 5 would be how many spores is in this syringe about. If we wanted to dilute it in half, kind of like what we're talking about, squirt it back out there, pull up another 5 cc's of water, squirt that in there, and just like that it was cut in half. And you know, in the process of doing this, that's kind of where the centrifuge tubes come on. And you know, you'd have this thing out, you'd have these guys out. Then I could go ahead and the last one would have been the 36,000 per milliliter, and this one would have been, was it 18,000 per milliliter? Now say we wanted to cut that in half. Squirt it back out. And that time we had about 10 cc's in the syringe, so we're going to have to take up 10 more. Make sure you're cutting it appropriately. Squirt that all the way out. Mix that. Pull that back up. Squirt it out, and now that's 9,000 per milliliter. And just like that, you can just really simply dilute your spore solutions and know exactly what you're working with. I see a lot of people talking about haploids and spore isolation and monokaryotic cultures, and they don't have a microscope, and they don't have a hemocytometer. I don't know, I'm not going to get into that right now. But, yeah, and once again, none of this was done sterilely. This was all just for fun. I'm going to be doing some actual dilutions for real projects, and I'll probably record those, and you can kind of see. I mean, really, it's going to be like you just saw for the most part, only I'd be using these micropipettes, sterile tips, all the sterile stuff. I'd be all clean. I probably wouldn't do it with the microscope here. Well, I'd have to actually. Yeah, I would have to have the microscope here, obviously. Yeah, but I guess I would make all my dilutions first before plating, obviously. So I'll do all my diluting one day, and then I'll probably come back and make the agar and plate them all out, and then it's just a matter of keeping a close eye on them and isolating them, making transfers, verifying them under the microscope as they're still in the dish before they connect with other colonies and transferring them. So I really don't know what else I could say. I mean, maybe I have a cool little fact. One more cool little fact. So if you had, okay, take one more step back. Let's say you had your on average one spore per cell. Okay, that's what we're looking for, kind of, if you have one of these guys. So you don't have one of those guys and you only have one of these guys. You know, say you have more faith in a syringes drop, because to tell you the truth, these are questionable. Like, real ones go for hundreds of dollars. I got this on Amazon for like 20 bucks. I should show you guys how small of a drop it can do. It's truly impressive, but maybe I'll save that for another video. Maybe I'll add it in. But real quick, so say you had an average on one drop or one spore per square, which gives you the 10,000 per milliliter solution, and you wanted to get it so just it was diluted even further to a standard drop. The quick little thing I figured out, the 1 to 499 thing, is actually one drop, or 50 microliters, to 29,450 microliters, or the equivalent to 29.45, about 29.5 milliliters. So you would take one drop to about 3 syringes, about. And all you need is this guy, and it would give you really, really close and accurate results that are really necessary for successful breeding, and even more so, like we might get into in another video, interspecific breeding. Not going to get too into this now, but I hope that some of this was helpful. I hope that, you know, even just seeing spores on a hemocytometer kind of encourages people to get one themselves and just kind of see what's really going on, because a spore is like a seed. Now, say you're, you know, if you have tomato plants or pepper plants, they interbreed all the time because people commonly have them in their flower gardens. So that's really what happens if you aren't isolating your spores, is they're back crossing and, you know, just intermixing with each other more than you'd like, basically. Because another big part of it is mycelium essentially breeds in the petri dish as well, to some extent. But I'm not going to get too far into that. But if you want to learn more about single spore isolation, I wish I could give you something. I mean, there's not a lot out there. There's not a lot out there as far as mushroom breeding, and I'm still trying to learn more and more about it. Just how far we can push our limits with these guys, and what kind of new cool options might be out there. If you look at what's done with plants, there's so much going on, and we are just touching the tip of this movement right now. So, hope everybody has a good day. I'm going to try to make more videos. Hopefully this one turned out okay. Cheers.